We're used to large language models predicting the next word or generating lines of computer code. Researchers have adapted this exact architecture to predict and output the sequences of A, C, T, and G nucleotides, the building blocks of biological life. A team from Stanford University and the Arc Institute used an AI model named EVO, training it to engineer entirely new biological entities. These are genomes designed from scratch. They have no natural evolutionary ancestors and have never been documented in the wild. The successful output of the EVO model was the digital generation and physical synthesis of functional bacteriophages, viruses that hunt and destroy bacterial cells. Because bacteriophages exclusively infect specific species of bacteria, they lack the mechanisms to enter human cells, posing no direct physiological threat to us. To understand the implications of this breakthrough, we have to look under the hood. We need to deconstruct how an AI model translates digital probabilities into viable physical organisms. The foundational architecture of EVO model directly mirrors the natural language processors that drive tools like ChatGPT. Instead of reading books, EVO was trained on massive, unstructured repositories of raw genetic codes sourced from viruses, bacteria, plants, and humans. Just as a language model learns that a subject, verb, and object form a sentence, EVO learns the rules of biological tokenization, recognizing how base pair sequences dictate protein folding. To establish guardrails, researchers explicitly excluded human-infecting viral genomes from its data pool. Once the system mastered this biological grammar, the researchers prompted EVO to generate novel structures specifically optimized to attack and neutralize bacterial organisms. By mastering this biological syntax, the AI system translates digital instructions into the specific functional blueprints of synthetic life. Digital generation is limitless, but moving those models into a laboratory introduces severe physical friction. Biological viability is governed by the laws of chemistry and physics, and many digital sequences collapse when physical synthesis is attempted. This funnel shows the attrition rate in the Stanford and Arc Institute study. In the first phase, the model generated hundreds of thousands of proposed viral sequences. The research team isolated 300 candidates from that massive output that exhibited the most promising structural geometry. 285 of those digital sequences were then chemically synthesized into physical DNA and introduced into a lab environment. Out of hundreds of thousands of digital designs, exactly 16 functioned as living viruses in the physical world. These 16 synthetic entities were fully functional, successfully breaching bacterial cell walls and hijacking the host organism to replicate their own DNA without any human intervention. The attrition rate from a digital prompt to a physical organism is massive, but the experiment proves that computationally generated sequences can achieve autonomous biological function. What is the specific medical utility of spending this much effort to synthesize synthetic bacteriophages? The researchers tailored the EVO model to hunt dangerous bacterial strains, specifically targeting organisms like Vibrio and E. coli. These targets were selected to address the accelerating global threat of antibiotic-resistant bacteria. Traditional chemical antibiotics rely on static mechanisms. Over time, bacteria mutate, outlast the chemical exposure, and evolve resistance. Bacteriophages act as dynamic weapons that physically hunt specific bacterial profiles and adapt within their environments. While bacteriophages represent a logical frontier for treating resistant infections, naturally occurring phages are limited by the same evolutionary constraints as their targets. This timeline shows the flaw in traditional phage therapy. If a natural phage is introduced, the bacteria can mutate to block it, continuing the biological arms race. The bacterial immune system catalogs the shapes of historical threats to recognize and block them in the future. The EVO model can bypass this by designing phages that have no precedent in natural history. Because the synthetic phage is novel, the target bacteria has no genetic memory of it. This allows the virus to bypass the cell's historical defenses. This computational approach allows scientists to generate new viral iterations on demand, aiming to outstrip the mutation rates of the bacteria. Generative AI decouples modern medicine from the timeline of natural evolution, providing a dynamic solution to antibiotic resistance. The exact precision required to bypass a bacterial cell's defenses can also be used to engineer novel human pathogens. The researchers explicitly flagged the biocontainment risks of their own methodology, noting that generating genomes raises important biosafety, biocontainment, and biosecurity considerations. When an AI generating text makes an error, the result is a hallucination, a factual inaccuracy on a screen. If a biological AI model hallucinates a novel genetic sequence that proves functionally viable in a lab, it becomes a physical pathogen. Because these biological hallucinations lack an evolutionary history, they represent a class of pathogens that current monitoring systems were never designed to track. The speed of AI development currently outpaces the structures of traditional health and safety regulations. Traditional biocontainment protocols are specifically designed to manage known pathogens, not the limitless iteration of completely synthetic organisms. While AI models attempt to restrict malicious outputs, researchers repeatedly demonstrate that models can bypass their own secure sandboxes. The final failsafe currently relies on the human in the loop, the physical scientist who decides whether to chemically synthesize the digital code. As AI masters the structural grammar of life, the challenge of engineering synthetic organisms becomes less about biological discovery and more about the scale of computational iteration and laboratory throughput. This shift moves biosecurity into an era where the primary bottleneck is no longer nature's complexity, but the speed at which we can design and test these digital genomes.